Hello, everyone, and welcome to today's webinar, Nucleic Acid Agarose Gel Electrophoresis Techniques, Innovations, and Common Errors. This webinar is part of the 12th Annual Precision Medicine, Genomics, Genetics, and Molecular Diagnostics Virtual Event Series 2024. I am Israel Gonzalez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, go to www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You will also notice that the speaker has provided a survey located on the left of your screen. This survey will be available during both the live session and the on-demand period. In addition to asking questions, we encourage you to participate in the 3 poll questions at the beginning, middle, and end of the speaker presentation. Click on your answer when the poll questions are displayed. If you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's get started. I now present today's speaker, Dr. Paulius Palaima, Product Manager at Thermo Fisher Scientific. For a complete biography on our speaker today, please visit the Presenter tab from the menu on the left of your screen. Dr. Palaima, you may now begin your presentation.
Thank you for that introduction. We will have several key topics that will be covered during this webinar, starting with the basics of description regarding electrophoresis and its history, and proceeding to best practices and common mistakes, as well as troubleshooting and how to avoid those mistakes. Electrophoresis has a very long history. Initially, it started actually as primarily for separating proteins, with a Tiselius instrument patented in 1930s. Nucleic acid electrophoresis took a while to catch up, so it was first applied in the 1960s, and then there were significant advances in the actual matrix that was used for the separation. So this included shift to agar instead of just using like starch or filter paper in some cases. And while this agar increased in usage over a period of time, a purified version called Agarose became the dominant version, the dominant matrix for nucleic acid separation specifically. And it kind of remained the status quo for a very long time, basically until today. There are other different types of matrices that are used also in nucleic acid separation, which is, say, polyacrylamide, but it's not as widely spread and it's more for a very specific use. It's worth mentioning that in 1998, while overall the use of standard Agarose electrophoresis was just using it in a bath, casting it yourself, in 1998 we introduced the e-gel Agarose electrophoresis system, which was dry precast electrophoresis, which completely shifted the paradigm regarding the nucleic acid separation. And later on in 2017 and 2022, we introduced 2 instruments that were, again, reimagining how we perform nucleic acid electrophoresis with the e-gel PowerSnap and the e-gel PowerSnap Plus. The principle of nucleic acid electrophoresis is actually very simple. So we have the preparation of the matrix, and as I mentioned, the matrix can both be agarose or polyacrylamide, but generally how it works is you have a negatively charged nucleic acid due to the phosphate backbone, and you have this separation matrix, which creates a sort of a net for simplistic purposes, from which, through which the nucleic acids have to move. And what that means is that if you apply a charge, those nucleic acids move towards the positive charge, so towards the anode. And they have to go through all of these different, like this molecular net that agarose and polyacrylamide can create. So larger molecules will have more difficult time to move through this matrix. So they will be moving slower, and smaller molecules will be moving faster. In that case, you get the separation by size. The uses of electrophoresis is extremely pervasive in all forms of in all types actually of molecular biology applications. So we have separation of complex mixtures of nucleic acids. So that's one big part, and then purification of nucleic acids of specific size. So the separation of complex mixtures is basically cloning. Total RNA isolation is a very popular one, and pathogen genome extraction is another popular one. So it's very important to understand that it kind of is applied in multiple different steps through multiple different applications that we currently use every time in the lab. So for cloning, it's very, very common to use cloning in a context of restriction digest analysis to see whether you have digested your plasmid. For total RNA isolation, it is also just to see how degraded is the RNA that you have extracted, whether you have two very strong bands of 18S and 16S RNAs. For pathogen extraction, this is also restriction digest, electrophoresis, and then we have genotyping following that, which also includes, to a certain extent, as an endpoint, electrophoresis. Purification is, in many cases, you will be probably familiar as applied in cloning to amplify your fragment using PCR, then extract it using electrophoresis out of the gel, and then use it for cloning later on. It can also be used for DNA and NGS. So for library preparation, to assess the size to isolate the proper size of your library that fits your application. And finally, amplification of genes for cloning. So this is basically what I mentioned previously, but it can also be in the sense of just general PCR analysis and, again, genotyping. Electrophoresis is an incredibly common molecular biology technique. It is also has like this traditional agarose workflow. It has been established over the decades that it's been in use. So generally, what you have is you have a gel buffer preparation. You have the gel and buffer preparation. But in which you melt the agarose. You cast it into a specific form. You make it with as many wells as you need it to be. Then we have sample loading and separation, which takes about 17 minutes. We have then staining, which is an optional step because you can actually add the dye into the agarose gel itself. But post-run staining has significant advantages when compared to in-gel staining. And then we have imaging and export analysis. Overall, this process takes around 2 hours and 20 minutes, as estimated by us. And if you really want to shorten it, you can use the Invitrogen eGel electrophoresis system workflow, which is very simplified in its sense. We have just 3 steps. So we have load, run, and analyze, because you don't need to prepare the gels, you don't need to prepare the buffers. You just load your samples, you run it on an instrument that is both capable of running it and imaging it all at once. So the total runtime can be just 18 minutes.
We would appreciate your participation by answering a few polling questions. To record your answers, click the options on the screen. Our first polling question is, which of the following applications involves the use of denaturing acrylamide gels in electrophoresis? 1. Purification of PCR products. 2. Gel shift assays. 3. Library preparations for next-generation sequencing. Or 4. Quantitative gel electrophoresis. I will now give you a few moments to select your answers.
So considering the multi-step process of pouring your own gel that we saw previously, let's see how we can achieve an exceptional gel. For this, there are several elements that we need to consider. So we have agarose gel that we want to have without the shadows of tracking dyes. We want it to be, for DNA to not be shifting in size. We don't want any smiling effects. We want our bands to be sharp. We want good resolution. We want the DNA ladder to be properly separated, as well as we want DNA ladder to have a proper density, so we can estimate the size of each of our fragments accurately. So basically, we want a gel very similar to what we are showing here on the slide. Naturally, this will require a lot of instrumentation. If you're following a traditional workflow, this will require a lot of instrumentation and a lot of reagents. So taking you through it, the most important ones is something that for the gel preparation. So you need to have a microwave to melt the agarose. Ideally, you would also have a heated stirrer, magnetic stirrer, that you can use, which will cool the agarose but still not let it set. And then you will definitely need the gel tank in which the actual electrophoresis will be performed. Then you need the power supply, which can produce the required voltage. You need an imager for your experiment documentation. And then you also need all the reagents. So we have agarose. It can be in the form of a pouch, like powder agarose, or it can also be in tablet form. We'll show you later. You need the stain. The most common stain is ethidium bromide for making your own agarose gels. And then you have buffers as well as water to dilute the buffers. This is much more simplified in the context of eGel precast agarose electrophoresis system, where you have all of these instrumentation just integrated into a singular instrument, and all the reagents that would otherwise be needed for the preparation of the actual gel is also already assembled for you in a very convenient shape of an e-gel. So, what are your options when considering the nucleic acid matrix, nucleic acid electrophoresis matrix? So, for pour your own segment, so these are the traditional workflow that you would use for making the gel yourself. You have a choice between powders and tablets. So, both of them have advantages and disadvantages. So for powder, it is generally quite inexpensive. It is very easy to store. It is, however, time-consuming because you have to weigh it out. Sometimes it can create messes because of shaky hands, and you just lose a little bit of your agarose every single time. And then you you do require all of the additional instrumentation that is associated with running these pour your own gels. For tablets, they kind of get away with you needing to weigh. So it's also takes up much less space. It is very easy to store, and it's very easy to adjust the agarose gel percentage that you wish to achieve. Because each tablet is just 1 gram, and you just pop it in, and then you have your agarose gel of the needed percentage instead of needing to weigh it out. For precast, we have the eGel agarose gels, which are fast. It's an integrated system and has no buffers associated with it, apart from the ones that you use to actually load the sample. So it's an extremely integrated and streamlined workflow. And then we have the polyacrylamide gels that are dedicated for nucleic acid separation. And what this means is that you have to have a separate instrumentation, as these are separate tanks, I would say, which are vertical electrophoresis. And in this case, you have extremely high resolution, up to 2 nucleotides differentiation. And it is also requires additional buffers and all the stuff that's associated with that type of electrophoresis. then this is where it becomes actually very important to understand what matrices you want to use, is specifically regarding the resolution and what is available for you instrumentation-wise. So if you don't have a vertical bath, vertical tank to run your electrophoresis, polyacrylamide gels probably are not going to be much use to you if you purchase them, because you cannot run them as a on a horizontal plane. So for majority of the cases, overall speaking, Agarose is a perfectly fine matrix to be used. It's the most commonly used matrix for a reason, and you can choose different percentages of it. It becomes much more difficult to prepare the higher percentage it is, because it becomes extremely thick and melting it becomes very problematic. But generally speaking, 0.7 to 2% agarose is very easy to handle. You can make it fairly easily. It still requires to go through all the steps of melting it and etc. But still, this is basically the bread and butter of most of molecular biology techniques, and you can see the separation distances that are associated with these type of agarose percentages here. And naturally, the less dense the matrix, meaning the less agarose you have in your gel, the higher the separation, the bigger the fragment that you can separate. It's just you have to consider that if you have small fragments, 0.7% gel is not going to be very effective at separating them, because they will just appear as a singular blob or a smear for you. For, say, fragments that are between 500 base pairs, you can choose 1% gel and 2% gel, but it's not going to work very well on the 0.7% gel. Same for the end part of it, where 10,000 or 12,000 base pair fragments will look as a singular band on 1 or 2% gel if you run it. Same for 4% gel. For polyacrylamide, it's not really meant for very large fragment separation. Again, the resolution is extremely high, but it's extremely high at very small fragment sizes. So between 6 and 20% polyacrylamide gels, you can have the highest possible fragment size of around 500 base pairs and the smallest fragment size of around 10 base pairs. And between them, you will have fantastic resolution. When considering which buffer you're going to use for your electrophoresis, you also have to consider what are the downstream applications that are following your experiment. If your goal is just to have a very nice gel with the fragment sizes of up to 2 kilobases, TBE buffer or trisborate EDTA buffer is going to be perfectly fine for your uses. But if you have downstream applications, say you're going to do cloning, or you're going to do some sort of a other type of related enzymatic reaction with your fragment, then TBE is going to inhibit those reactions, and your output is probably going to be very, very low. So for those applications, when you have downstream enzymatic reactions, tris acetate EDTA buffer is far more suitable. And for a reason, it's also the most commonly used buffer for electrophoresis overall. Then we have to choose the ladder. So if we chose our percentage, we chose our buffer, and the ladder choice will also depend on the percentage specifically of agarose that you're using, and the part of it that's going to dictate specifically what type of ladder you should choose is fragment size. And the next step is, which nucleic acid are you using? Are you trying to separate? For DNA ladders, for DNA, you should use DNA ladders. For RNA, you should use RNA ladders. That's basically the long and short of it. However, when you're choosing for the fragment size, you also have to consider the density of the ladder, how well it separates within your selected agarose percentage that you are running your experiment at. And to make sure that you have all of this properly selected, you should investigate whether the ladder separates throughout all of its range and it covers the sizes that you have. So here I have a quick example of a plasmid run, and this is just showing how plasmid in its different forms can migrate differently, so you also have to consider that. And then we have like the supercoil, we have the linearized plasmid, and we have the nicked plasmid. And it just shows that if you have a well-spaced ladder which separates throughout its range, you can very easily tell these things apart, given you have chosen the correct agarose percentage and buffers. To visualize your DNA, you will definitely need a dye, because generally speaking, you will not be able to see the DNA as it runs. And for this, the majority and the most commonly used dye, as mentioned previously, is etidine bromide. However, it has significant disadvantages regarding how you visualize it. You visualize it using UV light. So here what we propose is use cyber-safe or cyber-gold DNA stains, which use blue light transillumination. So this is particularly important in the context of also your downstream applications. Say for cloning, blue light will result in higher cloning efficiencies. But also it is safer for you because you're no longer being exposed to UV light. Additionally, different types of stains are also capable of, not all stains are capable of staining all types of nucleic acids. So double-stranded nucleic acids, say DNA, standard DNA, is very easy to stain, and most of the stain, because it's just majority of the molecular biology is based on study of double-stranded DNA. Majority of the dyes do stain it very well, and generally speaking, it's not really an issue. However, if you start working with single-stranded nucleic acids, say RNA or single-stranded DNA, then you have to really consider what stain you would want to use. And in this case, what we show here, say cyber gold, is capable of staining all different types of DNA and even forms of DNA. So it is important to consider that in your experiment. Additionally, then visualizing your DNA sample, as I mentioned, you have two different ways to stain your electrophoresis experiment, or your gel, to visualize DNA. And this is to add the stain during the preparation of the gel, so it would be within the gel, or actually do a post stain. So both of these have advantages and disadvantages. So if you add the stain into the gel, it can result in a shift, because the stain binds to the DNA inside the gel and it slows down its movement because it just becomes bigger, in a sense. It experiences stronger resistance while migrating through the agarose matrix. So as you can see here, while these are two exactly the same segments, exactly the same sizes, you can tell that, like, say, this fragment here is slightly higher than what this fragment is, representing a different type of size. You might be interpreting your experiment wrong if you're extremely sensitive to what the sizes of your fragments tested are. Additionally, you can also tell that these are not very nice. Sometimes they can impact the way that the separation works within the agarose gel. So you can see that it's not as nicely separated as what we can see in the post-run staining. So these are the disadvantages of using the in-gel staining. But it is, however, much more convenient, uses less dye and is a lot faster because you don't need to take additional step afterwards. Post-run, it is far more uniform. It is clearer staining. However, it requires an additional time after you have already run your gel. And if you leave it too long, you have a risk of having your bands become fuzzy. So the resolution of your gel is going to go down because it's just diffusion. So it's not recommended to have like a long post staining of your agarose gel. Also, there are additional things to consider when running, when staining, especially when you're staining inside, when you're using in-gel staining. So because the stain migrates in the opposite direction from what your fragment migrates, so your fragment migrates from, say, top to bottom, but your stain migrates from bottom to top. So that means that you will have some areas of your gel that are technically, like, it's called a shadow. And in those areas, the stain concentration is much lower. So your fragments will be much dimmer than what is, say, top of your, if you have fragments at the top of your gel. So this is a very important thing to consider regarding uniformity. So post-run, that's why it's a lot better, if you have a high range, say several fragments of different sizes spread out. Post-run staining is probably the best option that you would have, that you should, well, actually you should do. While in-gel staining will likely make the small fragments that migrate much faster appear much dimmer than the larger fragments that are at the top of the gel. Yes, and I touched upon this a little bit in the earlier slide, but generally speaking, linear DNA has a consistent and predictable migration by size. Relaxed nicked plasmid DNA is less migrates somewhat slower, as can be seen here. And the supercoiled DNA, because it is the most compact, migrates the fastest. So you also should consider this when you're performing your nucleic acid electrophoresis. So you have the proper resolution and the proper separation when choosing both, like, you know, the fragment, the agarose percentage, buffer, as well as you could differentiate them then using the proper ladder, which is which. So I covered what are the general considerations when making an exceptional gel. Now we can just look into some of the examples that we have prepared for you. So this is an example of a restriction enzyme digestion, and this is used like pour your own gel. So this gel was made by the scientist, and you can see the separation is really good. It's a very good gel. So we chose a very dense ladder, and we still got a very good separation between each of the bands. So we can definitely tell which fragments are of interest. As you can see, there are multiple different bands that you can see here. They probably digested it with several enzymes, as well as they are separated quite well. But this is why it's very important to have a good density of your ladder. And it's very important to have the ladder separated properly, because if you have a situation where you have performed a PCR experiment and your PCR is not specific enough, you cannot essentially design specific enough primers for your fragment of interest. You will get very similar results. So knowing what exactly is the size of your fragment becomes absolutely crucial, because you might have just smears, or you might just have multiple bands that have happened after many, many optimizations still, but you would be very confident in that this band is what I am very interested in. So getting back to this restriction digest, so generally speaking, for best practices within the context of restriction digestion, you should probably load about 30% of the sample, which is about 300 nanograms. So what this means is that generally speaking, restriction digest, many of the restriction digest protocols cap out at around 1 microgram per reaction. So 300 nanograms of that reaction should go into the electrophoresis just to determine whether you have a proper result. And that has reasons, namely because smaller fragments are less bright in electrophoresis than larger fragments. So you have to have significant amounts of both for them to be visualized at the same time properly without the larger or brighter fragments overshadowing the smaller, fainter fragments. There's also a consideration of the runtime. So when you are using large plasmids, longer runtime will definitely be very beneficial. Longer runtimes are also very good for just having a nicely run uniform gel like this without the smiley effect where the sides are moving slower and the middle is moving faster because there's just more electricity going through it. Sample loading die is another thing to consider. Sometimes sample loading dies can move can overshadow, but I'll show some examples later on of how this can look in troubleshooting specifically. Of course, if you're using e-gels, this whole process becomes much simpler. So when we have, say, if you're performing exactly the same experiment, you're using restriction digest, if you're performing exactly the same restriction digestion experiment using the traditional workflows, so you have 1 hour, 29 minutes to actually digest the sample, then it's 49 minutes of running. So we of running and preparing your agarose gel, and then we gave 15 minutes for visualization, because usually it's the camera or the system that you use for visualization is located in a different part where you have to bring your gel. For e-gel electrophoresis system, and especially when using the fast digest enzymes, this becomes 33 minutes total, which is about more than six times faster than the traditional workflow. So if you really want to save your time, and if you really need those experimental results quickly, or you just don't have any patience to perform all of these tasks that just waste a lot of time, you can shift to the e-gel workflow and it will be a lot faster. The benefits of your electrophoresis is basically the inherent high throughput, because you are the one that is controlling how many wells it will have. So generally, for high throughput electrophoresis, you can make up to 180 wells if you so wish, if you have the density of the comps that you can accommodate within that gel. And generally, you should probably go for about 1% gels for 400 to 8,000 base pair fragments. It still depends on the separation distance that you have, but these are the general guidelines. For 4% gels, it's 10 to 500 base pairs, so it's very high resolution of small fragments. So don't use very large fragments or even 1,000 base pair fragments at the 4% gel, because it just will not separate in any ways. There's also consideration of ladders. So if you're performing a high throughput electrophoresis run, meaning you have multiple comps and multiple wells very near to each other, so the distance of run is very short, you should also consider selecting the proper ladder that could accommodate that run and separate within that run. Because if we have, say, 1.5% or just 3% run distance, a ladder that is what I showed you previously with many different bands is just not going to separate, and it's going to look like a giant smear that is just going through the side of your fragment, and you will not be able to tell the size. Again, for buffers, same considerations, and for DNA fragment amounts, that still depends on how you're going to stain it, and still depends on how much you have, but generally, like, the guidelines are about 20 nanograms of DNA, because the stains usually have a sensitivity of up to 1 nanogram, so you will have significant intensity from each of your fragments to perform this run. Again, if you want to simplify the percentage-wise, and you have to prepare a lot of gels yourself, the tablets are a very simple way of just doing this, and you don't need to weigh out your agarose. You can just pop in the tablet and melt it, and you will get your desired percentage much quicker. High throughput electrophoresis workflow on eGel system is much simpler. You don't need to prepare a gel again. The gels have their own optimized ladders that separate properly over the distance, so you don't need to consider that. We have a selection of ladders that are specifically recommended with these gels. You run it on the same instrument as you would run it your smaller gel, say. So this is the eGel PowerSnap Plus. You instantly get your results, and without the need of exporting it specifically and like using USB keys and everything like related to the usually very convoluted infrastructure of many scientific institutions, you can export it very quickly to Thermo Fisher Connect, because this instrument can connect to the cloud. And in Thermo Fisher Connect, you can use additional analysis tools via iBite analysis software, and even have the intensity of the bands calculated for you. So it's very intuitive, very streamlined workflow that for high throughput applications makes your life a lot easier.
Our next question is, are you interested in a quote for an in vitro gen eGel PowerSnap system for nucleic acid electrophoresis? Indicate instrument of interest. 1, eGel PowerSnap Plus system, or 2, eGel PowerSnap system. Please take a few moments to select your answer.
So now let's go through the common mistakes and issues that you can experience when performing electrophoresis. As you can see here already in the cover slide, we have weird bands, we have low separation, there's blurriness going on, so all sorts of stuff that can happen just when you are trying to get your best result, but end up having some issues. So what are the most common issues with electrophoresis? So atypical band patterns usually caused by denatured DNA, incorrect electrophoresis conditions, so buffer prepared not well, damaged wells. Also, gel shift effect can happen during the atypical band pattern. Then we have curved DNA runs. So this is incomplete immersion of the gel in the buffer. So it's very important to make sure that your gel is completely submerged. Very low sample volumes. We can have also incorrect electrophoresis conditions as one of the recurring cases, meaning you might have just extremely high voltage that you set up. And there's also damaged electrode, which can result in these curved runs. Low intensity of band fluorescence, so insufficient DNA is the most common one. Insufficient or uneven staining, so in this case, what I mentioned before, you might be in the stained shadow, where it has migrated away from that area of the gel if you're staining inside gel. Then we have DNA run off the gel. This is one of the most common early mistakes. Once you do it once, you will never do it again, but it does happen a lot. Then we have diffusion in the gel. If you have forgotten about the gel for a long time, after you ran it, you can come back and it will be just very diffused bands. And then we have the DNA masking by tracking dyes or stain migration. So this is very similar to what I mentioned before. And then we have smeared bands. So we have DNA degraded, incorrect conditions, so wrong buffer concentration per se. Excess DNA loaded, so when you overload your gel, it will not separate properly and it will just have a smear. And then we have poorly formed wells, which can also cause migration issues and smearing of the bands. So here are some of the examples. So we have low voltage or long idle. So this is how usually it looks like. This is why it's very important to have to consider your voltage. So about 70 is fine. So 70 volts is a good voltage, but the run length will be longer. So for 70 volts, depending on what your separation distance is, it can be 40 minutes, it can be up to an hour, depending on how high of a resolution or separation you want to achieve. If you want to run it very quickly, you can run up to like 140 volts. However, you have to remember that if you run it too high, if your voltage is too high, the agarose gel actually can melt as the heat is not dissipated properly, and it just and it heats up. So in that case, you will also have issues with your run. So curved runs, this is what I meant. If you have too high voltage, your gel might look like this. So keep it around 100 volts, and you will be very fine, and it will give you a good separation at a decent time. We have also pore separation, so unsubmerged gel or short runtime. So short runtime is very easy to distinguish. If you run it for 15 minutes or less at 70 volts, you will not see significant separation, and it will be very evident just from the ladder, because the ladder is not going to have, the ladder won't have migrated enough. So in this case, it's just not gonna, you're not gonna get a good result. If you did not submerge your gel properly, you will also have issues very similar to this, where the gel is just, the electric field that's covering the gel is just not gonna be uniform. Atypical bands, so we have wrong loading dye that can happen, no denaturation or protein bound. So these are all issues that are specifically you have to consider when you are preparing your sample with the proper loading dye, and also what type of sample you have. If you have protein bound samples, that you know, expect issues with your electrophoresis unless you try to get rid of the protein that's bound to your samples. Smears and unresolved bands, so this tend to be overloading most of the time. However, it can happen when you have protein bound DNA or nucleic acids. Unresolved bands, so high salt concentration, so some dilution is definitely needed, because if sometimes you have reactions that have a lot of salts in them, that reduces the conductivity of the gel itself, and in that case, that increases the conductivity of the gel itself, and the bands will not be able to separate. If you have no detection, you might have ran the gel the opposite way. If you don't have a ladder and you don't have your samples, you probably have run the gel the opposite way. So you just switch the electric fields and the sample ran off. Or that could be your sample is not concentrated enough, so you need to add more of it, or you are using the wrong stain. So it might just not be able to bind to the form of DNA that you are using. So say for single-stranded DNA, in some cases, the stain is just not going to bind, depending on what stain you're using. So it's very important to know what form of nucleic acid you are testing, and that to make sure, or double check or triple check, that the stain that you are using is going to bind to that particular form of DNA. Incorrect band sizes, so gel shifting, that can happen, and I showed you before with the in-gel staining, that the sizes can, due to the binding off the stain, then using in-gel staining, that the actual sizes can shift a little bit, and the DNA fragment can appear larger than it actually is. So that is important to consider when you're performing electrophoresis. And then uneven staining, it can happen if you didn't prepare your gel properly, if you're using sometimes very large amounts of stain, or there's just dirty buffer in your tank. So it's important to change the buffer that you are running it in. So, additional failures of electrophoresis. So what I wanted to mention previously as well. So we have poor band separation, so wrong buffer and correct gel percentage. We have covered that in the previous slide. But what I really want to for you to notice, for you to pay attention to, is the selection of the tracking dyes. So a lot of these loading buffers have tracking dyes. So say bromophenol blue or orange J are probably the most common ones that I use currently. And these tracking dyes also migrate within your gel. And if you don't check what size of your fragment is and whether that corresponds to the migration distance or to the size that these dyes appear at, you might not be able to see your fragment because they will just overshadow your fragment. Because you see these white blobs, and in that case, you will just not be able to differentiate. So this is an important thing to consider when you are performing electrophoresis, so that your fragment lies somewhere in between these. In majority of cases, it is exactly that. It's somewhere in between them. In my long experience, I didn't have issues with this. So generally speaking, it's not a big problem, but if you have extremely small fragments or you have extremely large fragments, it is important to understand that this can arise and to tackle it accordingly when it arises. So overall, takeaways here is you have to think about the sample and what you want to do with it. So electrophoresis, again, you generally just separate it by size, especially in nucleic acids. And what are your downstream applications will dictate all of your reagents and conditions that you will need to select for your experiment. So the ladder, the staining dye, how much of the sample basically you need to load, and what is the Agarose percentage overall, and the buffer. Things to avoid, what we have is basically overloading of the sample. It must be avoided because you won't get proper separation. Damaging of the wells, this is... Why does it keep turning off? Jesus. Sorry, I'm not sure why does it keeps turning off. I don't know why. Okay, so things to avoid is overloading the sample because you won't get proper separation, damaging of the wells because you will have funky wells, as well as leaking of the sample for potentially cross-contaminating or just leaking of the sample overall out of the gel. We have very high and very low voltage, which has to be considered. Very high, you will have curved runs. Very low, you will have diffusion by the time that it runs. Different buffers in the gel tank and electrophoresis tank. These should both be the same. So both the gel and the electrophoresis tank should be prepared using the same buffer. Don't prepare, a common mistake was, don't prepare the gel with just water. You need to prepare it with a buffer. And improper amounts of electrophoresis running buffer in the tank. So just remember that at all times, your gel has to be completely submerged.
Our final question is, would you like to be contacted by our sales team to learn more about our suite of nucleic acid gel electrophoresis products and or the eGel precast gel electrophoresis system for your research? Yes or no? Thank you so much for participating in these poll questions.
So overall, nucleic acid gel electrophoresis is a very well-established method with close to 100 years overall of history, if we're considering early methods of protein separation. Pouring your own gels can be extremely challenging in some cases. And if you want to simplify your workflow and have less time-consuming workflow overall, eGel Agarose electrophoresis gels are a very good solution for that. And for best results, require understanding of what you are testing. So again, I just want to reiterate that your sample is very important to know what size of it is, what is your experiment goal, is electrophoresis just a middle step within your broader experiment. So consider that, because that will determine what sort of electrophoresis conditions you will need to set later on. We have a bunch of useful links for your information. So we have nucleic acid electrophoresis education webpage. We also have nucleic acid electrophoresis and blotting webpage, and nucleic acid gel electrophoresis and overview and history. So we have a full article covering electrophoresis history, which is very useful and very interesting to read. That covers in more detail what I said today in the first few slides. And finally, I would like to invite you to also check out the brochure for eGel precast systems, as well as all the other related material with eGel Agarose electrophoresis system, including our virtual tours, as well as our video manual for the eGel PowerSnap Plus instrument that just integrates and simplifies all of Poirot into just a singular instrument and a singular consumable. Thank you all very much, and have a great day. Thanks for having me.
Thank you, Dr. Palaima, for your time today and for your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank Thermo Fisher Scientific for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until May 15th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.